I'm Shanta Sabnisthomas.
And I'm Rebecca Allard.
This is our presentation for the Sanger Institute on malaria.
So, some background on malaria.
Malaria is a life-threatening disease caused by parasites that are transmitted to people through the bites of female mosquitoes. These are called malaria vectors.
In 2017, an estimated 219 million cases of malaria were reported in 90 countries. The deaths reached 435,000.
Also in 2017, 5 countries alone accounted for nearly half of malaria cases worldwide. These were Nigeria, the Democratic Republic of Congo, Mozambique, India and Uganda.
All important vectors bite from dusk till dawn. The intensity of the transmission depends on the parasite, the environment and the heat.
The treatments and preventions currently being used. Preventions include anti-malarial medication, these are some examples, mosquito nets and indoor residual spray with insecticides.
Treatments include blood tests to diagnose and anti-malarial medicines, as well as drugs to help the side effects of malaria.
Our budget and chosen projects.
We are theoretically given £20 million to spend on our chosen projects, so we have decided to put our funding on Project 10, which will cost £10 million and will help with parasite genome sequencing project, as well as funding of research towards gene drives and emergency funding for mosquito nets and repellent.
We wanted to focus on the research aspect of malaria treatments and preventions rather than the projects that involve practical applications in the present, such as supplying bed nets. These practical projects would supply short-term benefits on a small scale, perhaps in one country or state, and wouldn't eliminate the problem of malaria. However, some of the research projects we have looked into, such as Project 10 and gene drives, could potentially stop malaria on a larger scale long term.
Gene drives work by copying and pasting modified genes into both parents so that any offspring has a 50% chance of getting the modified gene. In 2012, a new gene editing tool became available. The gene drives based on this can be engineered into parts of the chromosome where there are gaps.
In September of 2018, the Imperial College showed that their gene drives could drive a small population of the mosquito Anopheles gambiae to extinction. The drive disrupted a gene called double sex that controls differentiation. If a mosquito had a copy of the drive, they passed it on to all their offspring. Females with 2 of the drive copied in, the number of these increases as the gene spreads throughout the population. They're sterile. Without a functional copy of double sex, mosquitoes cannot reproduce. This means that the mutations that could stop the gene drive targeting it are likely to stop the gene from working. Thus, the mosquito would be unable to reproduce and pass on the mutation anyway. This is a diagram of how it works.
The scientists at the Imperial are part of the Target Malaria Project, which is supported by Bill and Melinda Gates Foundation, as well as the Open Philanthropy Project Fund.
This project is already working in Burkina Faso, Mali and Uganda to prepare for the release of the gene drive, which will be introduced on top of a regime that includes bed nets, insecticide sprays and drugs for the infected. This ensures that even if resistance to the gene drive occurred, they could still break the cycle of malaria transmission by wiping it out in the trial area. However, gene drives do have implications. Modifying a species' genome could bring about unintended, irreversible changes that could affect our ecosystem. While no animals are known to survive solely on mosquitoes, mosquitoes are food for fish, bats and birds.
Genetic modification might also result in a further off-target mutation.
There is also argument on the fact that gene drives could be misused, for example for agribusiness and military interests.
If any genetically engineered mosquitoes are released into the wild, there could be an irreversible implication on the environment.
Gene drives are still only in the phase of laboratory testing, and it may be a few years yet until it reaches the phase of field testing. More tests have to be done first. While the gene drive worked well in small cages with a laboratory strain of Anopheles gambiae, this is no guarantee will work as well or at all in the wild.
We would like to fund gene drives because we believe that they have the potential to save hundreds and thousands each year by eradicating dangerous vectors that spread malaria.
Gene drives can also overcome the difficulty of stopping malaria in resource-poor countries.
Furthermore, according to the World Health Organization in 2017, progress had halted in 2016 and was the first time in 2 decades since the cases of malaria did not go down.
Finally, the breakthrough in gene drives could offer a long-term solution that eradicates malaria for good. Therefore, we think using 7 million of our funding for this research would be useful, since conducting tests and carrying out further research into gene drives will undoubtedly come with high expenses.
About Project 10.
It's run by a genome sequencing centre in the UK.
Project 10 aims to continue the research using sequencing technologies that will help to find the proteins on the surface of the parasite and determine their role and spread of the disease.
This research will be used to identify potential drug and vaccine targets.
Also, it will find the role of surface proteins in the red blood cells invasion.
It will make diagnostic tools for identifying parasites and host protein interactions. So, some background on Project 10.
Malaria research has been aided by new technologies such as genome sequencing.
By sequencing the first Plasmodium genome, it has revealed the genetic makeup of the parasite, allowing researchers to identify over 5,000 different genes.
This means that researchers have a list of genes to figure out the function of and the part they play in the spread of malaria.
Why we're going to fund Project 10.
This project will help to find potential drug and vaccine targets.
It will also show the role of Plasmodium proteins in the invasion of red blood cells.
It will also use technologies to find genetic variation that may affect the parasite's ability to invade red blood cells.
Diagnostic tools for the identification of parasite and host protein interactions can also be researched.
It is a great way to find out the long-term solution for the outbreaks of malaria.
Although it may take some time to find a vaccine or medicine, it could help to limit any future spread of malaria.
It also allows us to find out even more about the spread and growth of the pathogen.
This is how gene sequencing works.
First of all, the DNA is isolated from one of the cells and is separated and used as a template.
It's then mixed in solution with DNA primer, DNA polymerase, normal bases such as A, T, C and G, and modified bases.
These bases are separated into different mixtures so they can be read separately.
The mixture is then warmed so that DNA polymerase can work and make a new strand from the template.
To read the sequence, a researcher places the 4 separate mixtures into separate canals on a piece of gel. An electric current is sent through it, which makes the shortest DNA go the furthest, whilst the longest doesn't travel a lot in the gel. After processing to show the locations of the strands in the gel, it shows from the bottom up the sequence of the DNA.
This is a diagram of how it works. The pros and cons of Project 10. Some pros are that it helps to develop a vaccine which would have a long-term positive effect on a large scale. It will also find out more about how the pathogen functions.
Although, on the other hand, the cons include it is expensive and time-consuming, so may take a long time to find a solution, as well as if any of the DNA mutates, the drugs that have been found will no longer work.
Overall, this is how we'll divide the money. Firstly, we'd give 7 million to gene drives, because although it would take some time to see the benefits, it is a way to stop malaria in the long term for good.
We would also spend 10 million on Project 10 because we can find out more about the pathogen's genetic makeup, which would allow us to develop more effective drugs and increase our understanding of the disease.
Lastly, we'd give 3 million to preventions and treatments, such as mosquito nets, anti-malarial medicines and indoor sprays for insects. We would spend the remaining money on this because it would help control malaria spreading while the gene drives in Project 10 are being completed.
Thank you for listening!